we are really excited to have you here. And I will pass the virtual microphone to Carsten Carver, the ERA Chair holder in the Project WELCOME2, who has invited Stine to come to our institute, share her research, and learn more about us. Go ahead, Carsten.
Yeah. Thank you, Ivana. And of course, welcome also for me, Stine. So I guess you want to know a bit more who is Stine. Yeah, I know Stine since some 15 years. We were working together in a Nordic project, in Nordic European countries. They have also a special research project. We were together in a project called SysDiet, Systems Biology of Diet. Systems biology at that time was a buzzword you get funding. And so what I remember of Stine's career, she studied, in fact, there where she is now not only professor, but head of the department. So quite a career from young student to head of the department where she studied. And so you studied there, did the PhD there, and then you went for postdoc abroad, as everyone should do after PhD, and went to Strasbourg, nice Strasbourg, Johann Overk's group, and worked with nuclear receptors with LXR, if I remember correctly, so it's also joint interest with concerning nuclear receptors. Then you went back to Norway, to Oslo, first back to university, and then to a smaller town not far away from Oslo, Akershus, where you spent 11 years. Yeah? And then they called you back to your original department as a professor, and since 3 years, also department head. Yeah. Okay, and what else to say about Stine? We have together this NUTRIOME project. We're spending quite some time in writing it. Stine is coordinating it. We are lacking Parseval, who is the student from the NUTRIOME. I don't know where he is. Has he overslept? Anyway, we have 10 students in this Marie Sklodowska-Curie network that Stine is leading. In addition to science, we were also having joint interest in teaching, and we wrote already together 2 books, one on nutrigenomics, published a couple of years ago, and one on aging, published a few weeks ago, together with Eunike. Yeah? So, I guess this is everything what I can tell for the moment, and looking forward to your presentation.
Okay.
Stine,
yeah.
The floor is yours.
Thank you very much. Do you hear what I'm saying? Yes, I don't need a microphone. So thank you very much from the organizers and also from Carsten to inviting me here to Olsztyn. It's really nice to be here, and it's my second time in Poland. I visited Krakow almost 20 years ago, so I'm not so used to go to Poland, but I hope this will now start to speed up my frequent visits to Poland. So I'm going to talk about dietary intervention studies, which I am now primarily doing at the University of Oslo at the department. So as Carsten said, I am the head of the department, but I'm also a group leader at one of our divisions. And I will give you some examples of studies that we have conducted previously, and also say some words about what we are currently doing with some of the same studies, because we get a lot of data from these studies and we can use them for further analysis. And I will also talk about another EU project that I'm involved in, which is related to the exposome. So I have 45 minutes, and I hope I can manage to keep the time. So, but first, before I start going into the research, I thought I should also say something about the University of Oslo and our department. And the University of Oslo is the oldest university in Norway. It's not that old, but we are very proud still that we are the oldest university. It's from 1811. And I am at the Faculty of Medicine, which is the oldest faculty, from 1814. And the Faculty of Medicine is also one of the largest faculties at the University of Oslo. We are not longer the largest university in Norway, so that is something that we cannot really be proud of anymore. It's other universities, because there has been a lot of mergings of universities in Norway, so now we are the second largest university, but we are still the highest ranked university in Norway. And we have 7 faculties, and I'm at the medical faculty, and at the medical faculty we have 3 different institutes. It's the Institute for Clinical Medicine, Institute of Basic Medical Sciences, and Institute of Health and Society. And the Department of Nutrition is part of this Institute of Basic Medical Sciences. And at the Institute, we have 4 different departments, so nutrition is the second largest. We are about 80 people working at the department, and we are divided into 3 different divisions. And the largest department is this one, molecular medicine. And then we also have a department for behavioral sciences and in biostatistics. And for me, it's really nice to have all these 4 departments at the Institute, and we are located in the same building, all of us, because nutrition is really important, not only to understand nutrition, you also need to, it's really related to the molecular medicine department, but also to the biostatistics. They have a lot of expertise there that we can use and collaborate with when it comes to the statistics and analysis of the data. And also behavioral sciences is really important, also when it comes to nutrition, because what we really know is maybe we can do a lot of studies to show health effects of diet, but what people really need to do is to make behavioral changes. Okay, and at the department, we have 3 divisions. Very shortly, I will say this is the division of clinical nutrition, and I am part of one of 7 research groups, and my research group is one of these research groups at the division of clinical nutrition. And then we have a division of molecular nutrition, where there are 4 research groups, and then we have also a division of nutritional epidemiology with 3 research groups. So our department actually covers very many aspects of nutrition research, which is also really a strength that we have as a department. And we do now collaborate more and more together. Before we was maybe more in silos, but we are trying now to collaborate much more so that we can really use the synergy between us to be even better in research. I can just mention, because I will focus mainly on the division of clinical nutrition and the core facilities we have for doing dietary intervention studies. But I can also mention here that we also have very nice animal facilities at the Institute. Actually, now the animal facility at the Institute is now becoming the University of Oslo animal facility. So we are now we have actually merged all the animal facilities into one at our Institute. And also one of the research group leaders here is also the leader of the National Transgenic mice unit. So we also have a very nice transgenic mice unit at the department. As I said, we are trying to collaborate more and more together, and we have really worked the last years to try to find how can we create synergies between the different divisions, and what kind of research topics should we really focus on in the future. And these are the 3 research topics that we now have agreed upon, and we are now trying to more work closer together, all of us from the different divisions. And it's the healthy aging, which we see as really, really important for the future when it comes to diet. Obesity, lipids, and cardiovascular disease. This has been for many, many years the main topic that we have been focusing on, and also my own research focus has been on cardiovascular disease and lipids, which I will come back to later when I show you the results and examples from my intervention studies. But also cancer and malnutrition is an important part of the activities and an important research topic at the department, which I'm not involved in at all for the moment. Okay, so I will just say a few words about the division and the core facilities we have for the performing diet intervention studies. As you see here, we have a very nice, shield? I don't know the English name right now, but you see here it's Center for Clinical Nutrition. And we have very nice facilities for the participants that are coming to our intervention studies for blood sampling. We have very nice measurements for measuring body compositions. We have DEXA, very advanced DEXA instruments, and also BIA instruments for measuring body composition. We have very nice rooms for where we have the possibility to, of course, perform a lot of different anthropometric measures. We can also use it for grip strengths, and to study grip strengths, we have axillary possibilities to measure physical activity. That we can do different kind of physical tests. measuring blood pressure and so on. So it's very well equipped to be able to do a lot of different measurements of the individuals that we are having in our intervention studies. I primarily work mainly not with patients. I work mainly with participants that I recruit via social media from the general population. But we also have an outpatient clinic at the department that is not ours, but it's the university hospitals, because we are very closely located together with the hospital. So, and that is primarily cancer patients that we have that is coming to this outpatient clinic, and where there's also a lot of clinical nutritionists working. So we also have, that is also a very good thing with us, that we are so close to the university hospital. And we also have kitchens, which we use for training. We are also responsible for a 5-year master program in clinical nutrition, but we also train the medical students in nutrition at the faculty. And so we have a kitchen that the students can use, but we can also use it when we're doing our intervention studies. And lately, the newest thing that we have invested in is in indirect calorimetry rooms. And these are quite unique rooms. I know there are rooms in Germany, and it's rooms in the States and also in the Netherlands. They are a little bit different, but this gives us the possibility to really measure oxygen consumption and also CO2 production, and we can really measure the metabolic rates in subjects. And we have 3 different rooms, and for the moment, it's this one that is validated, where we can measure resting metabolic rate, and people can stay here. We can do diet-induced thermogenesis measurements. And we also have an exercise room and a room where people can actually stay overnight. But these are still not validated, these 2 rooms. It's quite demanding, and these were just finished before COVID, so we of course had to stop everything. But this room is now up and running, and we can use it for our experiments. Yeah, and this is just the measure, how we can measure all these, the gases and so on. Okay, so this was a little bit about the institute and the department where I come from. And now I would like to talk about my own research, and I'm particularly interested in diet as a powerful prevention strategy. And I'm mainly working with cardiovascular disease, but of course cardiometabolic diseases, also including diabetes type 2, is also of course very important when it comes to diet and the preventive effect of diet. So we know that cardiometabolic diseases, we can think about it like 3 main components that is causing the diseases. And of course, lifestyle is one of the major players, not only diet, but also, of course, smoking, physical activity is important. We know that the environment are important, and I will come back to this later. And when it comes to the genes, we know actually that maybe the genes account for maybe only 15% of the reasons why we get the diseases. And that is, and that makes, for me at least, the interest when I'm working with diet, to know that it's not directly the genes, but it is how the diet can influence the genes. That is important to understand how diet can influence and prevent cardiometabolic diseases. And diet is, of course, important. You know all about diet and gene regulation, so I will not go much into details to this, but Karsten just mentioned it, that there was one receptor that we can have in common, the liver X receptor. But my focus for when I was also a postdoc, but also as a researcher now and as a professor, it's been primarily on fats, because we know that fatty acids and also cholesterol can act as ligand for transcription factors, or they can be important for activating transcription factors. So this gives me a rationale for trying to better understand how fat and fatty acids can affect health and promote health and to avoid diseases. And I was primarily, before I did my, before I went to the university, this Akershus University College that Karsten mentioned, I was primarily working with mice and cells, very much with experimental research on in these model systems. But since 2004, I've been primarily working with humans. And for me, it's about really understanding how diet can influence our health by acting as we know that the food is rich in nutrients, which can have different effects on both the epigenome, the transcriptome, the proteome, and the metabolome. And in my research, we have primarily been focused on the effect of nutrients on the RNA expression using different techniques, primarily transcriptomics, which I will come back to. And lately, we have also started to focusing more on the metabolites, because also, of course, it's really important, the effect of nutrients on the epigenome and the RNA and the transcriptome. But many of the genes that are regulated by nutrients, they are also encoding for proteins that are binding proteins for nutrients, or they are important for uptake of nutrients. They are cofactors, they are enzymes, very important for the metabolism of nutrients. So, of course, all the effect on the DNA and the RNA and the protein will also, of course, in the end, affect the metabolites that are present. So that's why we are also now focusing more and more on the metabolomics and not only the transcriptomics in our dietary intervention studies. Lately, also the effect on the gut microbiota has been very much interesting. We are now starting to also include this in our interventions, but we have not been there at all. So for my talks today, I will mainly talk about transcriptomics and metabolomics. Okay, so the first study that I would like to talk to you about is about fat, and it's about replacing saturated fat with polyunsaturated fat in the diet. Because I don't know how it is in Poland, but in Norway we really have a struggle when it comes to the population and how they are eating fat, their fat intake. It's very, very well documented that that saturated fat increases the risk of cardiovascular disease. And it's also very nicely documented how you can replace saturated fat with polyunsaturated fat and see the health effects of this replacement. So increasing, but when you look at this is the national data from Norway, when we look at the diet, Norwegian diet, we still eat too much saturated fat. The recommendations is below 10 energy%, but in Norway the intake has been quite stable, and it's for the moment around 14 energy%. And I don't think this is unique in Norway. This is it's a very I think in many European and also worldwide, this is a common problem. And the intake of polyunsaturated fat, they recommended intake should be between 5 and 10 energy%. But in Norway, it's been around 6 energy%. So it's still below the recommend, or it's within, but it's still a possibility that you can actually, if we could manage the population to exchange some of their habits, change their habits so they could consume less saturated fat and increase the intake of PUFA, this could have a good beneficial effect. So how to do it? It's really, really difficult. We have struggled. This is one of the main struggles we have. So what we did some years ago was that we wanted to see what will happen if we replace a few commercially available food items in our diet with the similar food items, but with different fatty acid composition. How is this possible, and what kind of health effects can we be able to detect? So we did this study, now it's more than 10 years ago, where we, in a collaboration also with the food industry, we started this study. And the aim was to investigate the health beneficial effects of replacing saturated fat with polyunsaturated fat using what we called a food exchange model among healthy subjects with moderate hypercholesterolemia. And the primary outcome was total and LDL cholesterol, because we know that saturated fat will influence cholesterol. So what we did, we designed this study, and we recruited people from the municipality. We used different kind of social media and also advertisements in the newspapers to find people that had a little bit high LDL cholesterol. It should be above 3.5 millimolar per liter. They should be healthy. And the age group should be between 25 and 70, and they should have BMI, so they should have normal BMI, but also be overweight. So we had a quite range of BMI between 20 and 35. And in total, actually 700 people contacted us and wanted to participate, but we had a lot of different inclusion-exclusion criteria, so there was quite many that we had to exclude, but we ended up with randomizing 115. And this is a quite large dietary intervention studies when it comes to this kind of interventions. And then what we did was that we, of course, randomized the group into a control group and then to an experimental diet group. So this we decided as a double-blind randomized control trials because all the foods that they received, they were packed in packages so that they couldn't really know what kind of products or brand of products that they were eating. And we had a run-in period before they were randomized, and in this run-in period, everybody was eating the control diet products. And I will come back to these, what kind of products these were. But the overall goal of this was that we were able to replace 6.5 energy% saturated fat with the equal number of polyunsaturated fats. So we really we managed that in these dietary intervention studies. So how did we do it? What we used, it was, as I said, food items that the Norwegians are normally eating. And it was based on bread. In Norway, we have bread for breakfast and lunch very often. So we said that the minimum amount that the people should eat was like 3 slices of bread. It was a portion of cereals. It was butter or margarine-based butter, and it was also oil that we could use for cooking. It also included creme fraiche, mayonnaise, and also liver pâté. And the reason why we chose these products was because there was a brand produced by an industry partner, Mills, that had these brands made, these food items made. And these food items were made primarily that they had replaced saturated fats they used instead oil, sunflower oil and rapeseed oil in the production. So they replaced the saturated fat with more omega-6 fatty acids. So the food that the experimental group received were these food items, which had then a much higher content of omega-6 fatty acids compared to the control items, which were the same items, but then with the more produced by the original way. And we used the amounts, we based it on the nutrition surveys in Norway, which is also where my department is responsible for these nutrition surveys. So these amounts is what we know the people would be able to eat based on what we know that the Norwegians are normally eating. And what the fatty acid composition, as I said, the main thing here is that the food, it was similar food, it's just the different fatty acid composition. And in the control group, these products, as you see, were having a less content of linoleic acid, omega-6 fatty acid. And it was a much higher content of the saturated fatty acids. And it was basically all of them, both the myristic, palmitic, and stearic acids. And these are also the fatty acids that are known to influence cholesterol. The MUFA, the monounsaturated fatty acids, were the same, so it was no differences in the intake of the monounsaturated fatty acid. It was primarily the change between the PUFA and the saturated fatty acids. And what did we see? Actually, what we were able to show was that we could actually reduce cholesterol, both LDL cholesterol and total cholesterol in the experimental group compared to the control group. And for us, this was really, really nice findings because it really shows that it is possible by just making simple changes in your habitual diet, choose the correct products. You can even, and then you don't really need to make a lot of changes, as you could see. And still you can have a very, very important biological, it's really an important reduction in cholesterol when you do these changes. So this was something we were really happy to see, that it's really possible. Of course, this was only for 8 weeks. We don't really know if it could be possible to do these changes for a longer period. This is what we cannot say anything about. But of course, if these people were continuing eating the experimental diet group food items, which are commercially available, it could really have a health beneficial effects also for many more people. Okay. We also measured some of inflammatory markers because there has been some concern that if you increase the intake of omega-6 fatty acids, it may have an influence on inflammation. But at least when we measured some of the inflammatory markers in circulation, we could not see any changes in these inflammatory markers when we compared the effect between these two groups. Okay. And then we went further to try to really maybe more understand mechanistically how can we understand the health beneficial effects of replacing saturated fat with polyunsaturated fat. And we did we worked with that for some years. And I will just very briefly mention these results as well. And we use for understanding the mechanisms, we normally use the PBMCs. These are immune cells in the circulation, and they are easy to collect. We can just use specific tubes and when we are collecting blood. And so it's not really a very invasive, it's not really invasive if compared to if we, for instance, were going to take biopsies, for instance. So the blood is a very good system. And we also know that nutrients are able to affect gene expression in PBMCs. And also the PBMCs, which are mainly consisting of monocytes or lymphocytes, we know that these cells are very also important for the atherosclerotic process. So for us, it's also a very important model system since we are interested in understanding the effect of diet on cardiovascular disease risk, as atherosclerosis is the driving force for many of the cardiovascular diseases. So what we did was that we collected PBMCs before and after the intervention in both groups, and then we compared the expression profile in the experimental group and the control group, and we looked at the differently expressed genes in these groups. And here what we could see was that in the PBMCs, we could see that it was more than 1,100 genes that were differently expressed between these two groups. And here you just see this volcano plot that this is, and this is also based on the p-values, and you see that these are the genes that are downregulated in the experimental group compared to the control group, and these are the genes that are upregulated in the experimental group compared to the control group. Of course, these are just nominal p-values. It's not adjusted for multiple testing. But what we normally do is that we always use these gene transcripts that has a p-value below 0.05 into pathway analysis to try to understand the interpretation of the results. But when we adjusted for multiple testing, and here this is a lot to discuss about the threshold of these adjustments. But in nutrition studies, it's quite common to have not be too conservative when it comes to the adjustment for multiple testing. And here, what we could see was that there were 14 differently expressed genes that were differently expressed between these two groups. Some were downregulated and some were upregulated. And what does this really mean? Well, it's not so easy always to interpret the data, because some of these genes are sometimes genes that we don't really know the function of. But at least when we looked at some of these genes that were differently expressed, we could see and try to see if any one of them could be associated with cardiovascular disease. We could see that at least 2 of these genes were involved in regulation of blood pressure. 2 of the genes were also important for proliferation of smooth muscle cells, and also 1 of these genes were also involved in the LDL receptor protein folding. So at least it could give us some new ideas about what is really the health beneficial effects of replacing saturated fat with polyunsaturated fat when it comes to the risk for cardiovascular disease. We also correlated these changes to LDL and both total cholesterol and LDL cholesterol, and some of these were also correlated, both positively or negatively, to the change in LDL cholesterol, at least giving us some hints that they might be also directly involved in the regulation of, or explaining the effect of the reduced effect on total cholesterol and LDL cholesterol. And we also did pathway analysis, and here we used all these genes that were differently expressed with a nominal p-value, and mainly the pathways that were enriched in the experimental diet group compared to the control diet group were in the immune response, chemotaxis and cell adhesion, and also signal transduction. And of course, these are also very important processes when it comes to atherosclerosis. Okay, so this was the gene expression. We also, as I said, we have been also interested to perform metabolomics. And we have used two different approaches when it comes to metabolomics. And in this study, we used NMR, and we used a commercial lab in Finland. It's the Nightingale platform, or Nightingale lab. Nightingale Health, I think they are named now. And with this NMR analysis, we can get a lot of data on different kind of lipoproteins and lipoprotein particles. And these are, of course, important in the transport of lipids in the circulation, and they can give it much more detailed information about the sizes of the lipoproteins and also the composition of the lipoproteins. In addition to that, they can also measure some amino acids and also some other metabolites. But what we primarily focused on, and what we could really confirm and see in our data, was it was mainly here is the very large density lipoproteins in different sizes, and it's also the LDL lipoproteins with different sizes. We could really see that these were reduced in the experimental diet group compared to the control diet group. So actually we could really see, and these are the main atherogenic lipoprotein particles. So we could also somehow confirm the data that we knew that this intervention changed the cholesterol, total cholesterol and LDL cholesterol, but they also influenced the level of the most atherogenic lipoprotein particles as well. So this was very nice to see. We then also, in collaboration with the University of Copenhagen, with Lars Dragstedt group, who is very, very good in untargeted metabolomics, and here you use mass spectrometry. We also looked at the metabolites in serum in these two groups, and what we also could very nicely show here was that when we were using this more untargeted, it's more exploratory metabolomics analysis, we could see that the red group here, which is metabolites, and they are primarily from the experimental diet group. You see there's a very nice separation of these metabolites, and these were mainly phospholipids with, of course, unsaturated fatty acids bound, enriched with polyunsaturated fatty acids. And then you had these green ones are the metabolites that were measured from the control diet group, and these were mainly phospholipids with saturated fatty acids. So we can very clearly show that we were in this intervention having a very clear separation on the metabolites, and it was mainly due to the fatty acid composition of the different phospholipids. We also wanted to see if there was any associations between these diet-specific metabolic profiles and the cardiometabolic risk factors, and what we could again show that there was a very significant positive correlation between the control diet group and the cholesterol, both the LDL cholesterol, triglyceride, and total cholesterol. So showing that these phospholipids with saturated fatty acid, they could increase these LDL cholesterol, triglyceride, and total cholesterol, while the experimental diet were significantly inverse associated. And it was the same risk factors, but again, this was the phospholipids that were particularly rich in the unsaturated fatty acids. However, when we did mediation analysis, we could not really say that it was the metabolites that was driving this effect on cholesterol. It could maybe be more that it was just reflecting the diet, or it could be that we don't have sufficient power enough to really show that it was really those particular phospholipids that were driving these changes in the cholesterol levels. Okay, so this, just to summarize this study, so we could show that replacing saturated fat with polyunsaturated fat is able to modulate the gene expression in PBMCs and also pathways related to CVD risk. And we could also, by applying metabolic profiling to this RCT, where we could also extend our knowledge on the biological and molecular effect mediated by fat quality. And we also see that changes in dietary fat we can see that in the metabolome, and it can also confirm compliance to the intervention. Of course, we really need to do further studies to identify metabolites that could explain better the clinical outcome, because, as I said, for the moment we have not been able to do that. Okay, then I want to shift a little bit gear and talk about another study that we have been conducting, also because we are interested in fat and cardiovascular disease. And I will give you an example, and this example is from a meal study that we did also some years ago. And this was also a study where we were collaborating with the industry. This was an innovation project together with, which was funded by the Research Council of Norway, because they have, in Norway, the Research Council is supporting projects where we collaborate together with industry. So the industry is paying half of the funding, and then the Norwegian Research Council is supporting us with half of the money. It's a very nice funding possibility to be able to work together with the industry. And we think that is important, in particular in the field of nutrition, because it's very difficult to get funding for nutrition studies. And this is one way to actually get funding to do nutrition studies. So in this study, we were particularly interested in dairy products. And of course, dairy products is a very interesting food group. When we talk about dairy, we very often think about it as one food group. But dairy products is a very large variety of products. And you have, for instance, you have fermented and non-fermented products. And you also have, of course, the fatty acid composition differs in these products. We know that in general that dairy contain a very high amount of saturated fat and is linked to increased risk of cardiovascular disease. But there is also some debate about this. And in this study, we were using 4 different products. So it was cheese and sour cream, which are both fermented, and it was all butter and whipped cream that are non-fermented. But the products are these 3 products are very similar when it comes to the fatty acid composition. And cheese is, of course, a little bit different because it also have a very high protein content and calcium, protein content in general. I will show you this later. And here the aim was to really look at triglyceride increase, because triglycerides are important. Fasting triglycerides is a risk factor for cardiovascular disease risk, but also the postprandial increase in triglycerides is also a risk factor for cardiovascular disease risk. So here we did a study where we wanted to investigate how high-fat meals containing similar amounts of fat but from different dairy products, how that can affect 6-hour postprandial triglyceride response. To better understand how nutrient composition, different food structures, and industrial processes in dairy products may impact metabolic responses. I see the time is running, so I will very shortly this is the design of this study. This is also with lean, but also lean and overweight subjects. They were healthy and they were between 18 and 70 years old. They were also recruited from social media. And they were all going to eat all these 4 meals, but in a fixed order. They were randomized, so they were either eating butter, cheese, whipped cream, or sour cream. One group did that, and then the others, they were all having this fixed order, in which order they were going to eat the meals. So all ate the same meals. It was 3 to 5 weeks between these test days. And then we were sampling quite frequently. We had fasting samples, and then we sampled for 2, 4, and 6 hours, and we could then measure triglyceride and also a lot of other measurements to see what happens in the postprandial phase. These were how the meals looked like. It was 3 slices of bread with then 60, I think it was 60 grams of either butter, cream, whipped cream, or sour cream. Everybody actually liked these meals, but we had a quite high drop-off after the first meal, because I think it's a bit challenging also to eat these meals. But what I want to show you is the fat. The fat content was very much the same in these 3 meals. But of course, the fat energy percent was a little bit different, because it was particularly with the cheese, because the cheese was also very high in protein, as you see, and it was also having a higher calcium amount. So it was a little bit different, yeah. But when it comes to the results, what we did see was that when we looked at the triglyceride, which was the main outcome, the triglycerides very often increase. That's a very normal pattern, increase after 2 hours. And it was mainly the sour cream that actually had a very high increase after 2 hours, but then a very rapid clearance, while the other products were behaving more similar and different. So there is something with this sour cream. We don't know yet what it can be, but that may have an increase the triglycerides, increasing this increase faster, but also that you had a faster clearance. Yeah, so that was the main finding. We also have a lot of other data from this study, which I will not go into, but I want to say further is that triglycerides are really, really interesting, because we know that there is a large differences in response when it comes to triglycerides and response to, for instance, omega-3 fatty acids, which we also have been very interested in doing interventions on. And this large differences in response is, of course, also something that we are very, very much interested in when it comes to precision nutrition, to really understand these differences in responses. And we do believe that maybe based on how people respond and why people respond differently, maybe we will also need to have different kind of dietary advices for these people. But this is just to show you one example from one study that we performed many years ago on fish oil intervention, just to show you the concept that this is the change in triglyceride. It's in percentage. And what we could see here was that there was a lot of people that actually had a decrease, which you would expect from fish oil, a reduction in triglycerides. Then there's a lot of people that don't respond at all. And then you actually have a group that actually are having more an adverse effects, so that increases their triglycerides. For us, this was a bit, it looks strange, but it's really what is normal when you do intervention studies with fish oil intervention. And this is just to show you from another big study in England, the FinGen study. It's very much the same. They also gave fish oil to subjects. And this is also what they also observed here, that there is a large group of responders. There's a large group of people that have this decrease in triglycerides. And then you have a large group of people that hardly have any, some of them hardly have any effect. And then you also have somebody that actually increases in triglycerides, just like we saw as well. And many of you probably know this PREDICT study in UK. It's a big study in England where they also looked at triglyceride responses. And this is they very nicely show the large variation in triglycerides when it comes. This is about 1,000 subjects. So there's a large, we know this is a large variation in triglycerides. And there is also a variation in how you respond. And in this PREDICT study, they also, so this is the reasons then why we now are starting to think about this one diet fits all to more the right diet to the right person to the right time. So what they showed here was that there was the serum lipids markers and the serum glycemic markers. They are really the main markers that can explain this variation. And I mention this here because right now, one of my PhD students in this Nutriome, what she is really working on, she's now working on this data from this dairy from our study here, to look at the metabolic response curves, because it's really, really fascinating also when you start to look at the individual data to see how people, the response curves are very different. And we are trying now to cluster the subjects to see if we can see if there are certain clusters of individuals that respond differently, not only to the increase in triglycerides by itself, but really to see the metabolic response curves. And what Jaing in my group, I'm not going to show you the results, just to explain for you. She had done this analysis. She has found clusters, and it's really, really fascinating to see that some subjects here, and the strength of this study is that we have repeated measures. We have 4 different meals, so we can do more sophisticated statistical modeling, is that some people actually hardly have any increase in triglycerides at all. And then you have those that have this expected pattern. And then you have people that, which you have an increase in the peak, and then they go down again after 6 hours. But then you also have subjects. You have a cluster where subjects, they stay high, they don't go down again. And to try to really understand why is that happening, what is characterizing these subjects that has these different metabolic response curves. That is something that we really now are looking into, which I think is really, really important, because that can also be important when you are designing new studies, diet intervention studies, to see if we could group them differently based on their metabolic phenotype, and it can also help us to understand better the effect of diet. I see that the last thing, just very briefly talk about, last thing before I end, is to change a little bit. I will not talk about diet so much. I'm trained as a nutritionist, so my focus all my years have been in nutrition. But I must say that when I'm been now part of a large EU project where we are looking at the exposome, you get really fascinated about all the different factors that are actually able to affect us. It's not only diet or physical activity as said here, or drugs, smoking, alcohol use, sleep behavior. This is maybe where my focus has been, on the lifestyle. But you know, it's everything that we are surrounded by can affect us. And this, I think, is a very nice paper, and it's very nicely showing actually how the whole environment around us is affecting our health. So and when we look at it, this is another nice picture which I like, is also to talk about the specific external environment and the general external environment, and how these can interact with the internal environment. And in the internal environment is, of course, the epigenome, the transcriptome, the metabolome, and the proteome, and everything, and how that again can affect the health risk. And I think these are more the specific external environments that we can actually do something about ourselves. This is our choices. By our own choices, we can make changes to affect the internal environment and our health. But then it's also really important to think about the general external environment, which is actually where we are living, the air pollution that is surrounding us, the green space, the blue space, all these things is also affecting us, and can also affect the internal environment, and then again affect our disease risk. So in this, we are now part of a large EU-funded project, and this is to study interactions between the whole environment, lifestyle, and cardiometabolic diseases. And it's 18 partners, and it's 8 countries. It's headed by the University of Oslo in Finland. And I will not go into too much details, but our main task in this project is that we are responsible for the transcriptome analysis. And in this research, in this consortium, there is a lot of access to birth cohorts and also cohorts on adults and elderly. And of course, there is a lot of different outcomes to look at. So I will give you now just one example from one of the studies. And it's in adults, but we are also working with the children in 2 birth cohorts. And the data that we see is quite similar. And of course, why are we interested in For me also, I'm always been thinking about diet as really the main risk factor for cardiovascular disease, but also air pollution is actually one of the main risk factors, both in male and females, about cardiovascular disease. It's air pollution you see here, and it's of course it's also driven by the cardiovascular disease, but also of course a lot about the lung diseases and cancer and so on. And these are female and males, it's very similar. What is really interesting is that at least what we are working now with is air pollution and its particulate matters, and these are in different sizes, and this is what you get from the traffic, for instance, the traffic. And what is fascinating is that many of these particles, they are affecting the same mechanisms as how we know that nutrients can affect it. So this can be an interplay between nutrition and air pollution. So one idea also you can think of is if you live in a very polluted area and have a very poor diet, you are more, you will have a higher risk for disease if, like, you were living in a very polluted area but having a healthy diet. You can reduce the risk, for instance, from living in this polluted area. This we don't know anything about yet, but that's how we can think about it. And so we used, it was a postdoc in my group, Amanda. She used the same study that I showed you before. We used people that we had for this screening. It's not a big group because we had to ask for permission again from the ethical committee to actually include them in this longitudinal study. So it was 98 that responded back that they were interested to participate. And what she looked at was also gene expression. And what we could see that was 19 genes that were significantly associated with exposure to particulate matter 2.5. That is the really smallest particulate matter. She also looked into nitrogen oxide. She looked into built environments such as access to green space and blue space. And we did this by I'm not an expert in this, but we used the addresses to the participants, and we use geocoding use the addresses, and then we had data on the pollutions from different sources, which are available in this longitudinal project. I don't know all these details, but we were able to actually use the addresses to these subjects to find out what they were exposed to, how far. And then we could see that there was 19 genes. This is just some of them that were negatively associated with air pollution. And many of them were also related to lung function. And we also repeated this in a bigger study, a Rotterdam study. And interestingly, we could see overlap in the genes that were associated with air pollution, but they were not really in the same direction. So this is also a bit of debate now about the level of exposure, how that can affect these genes. But anyway, it's very similar also to what we see in children, and it's primarily PM2.5 that is associated to gene expression, not the other environmental factors. So yeah, so this was just this conclusion, and yeah, now I just want to end. I see the time is running, so I just want to thank all the collaborators and all the people at the institute or the department that is working with me, and also all external partners. Carsten, when you were in Kuopio, we were collaborating on the NUMA study, and now we are collaborating on the NUTRIOME study, and of course also longitudinals I have to thank. Yeah. Okay, thank you very much.